Hello, I'm Ashley Obish, and today I would like to talk about clustered regularly interspaced short palindromic repeats, or the CRISPR-Cas9 system. Originally, CRISPR was used as an immune system in bacteria to fight phage in the cell. This is why we created a new and improved form of CRISPR, or CRISPR-Cas9. Cas9 represents a CRISPR-associated enzyme 9 that uses CRISPR sequences as a guide to recognize and cut specific strands of DNA that are complementary to the CRISPR sequence. These are the scissors. In the cell, this technology can be used to prevent mutations and diseases by providing a homologous strand of DNA to repair the broken DNA. This is usually done through HDR, or homology-directed repair, which causes the DNA to go through ligation and repairs the DNA without harming it. The other process is NHEJ, or non-homologous end joining. This is when the DNA is cut but not replaced with a complementary strand to repair it. The cell tries to repair itself through trial and error. It is extremely error-prone and can lead to mutations within the DNA, especially because it skips the process of ligation. This tool may be the answers to all questions regarding whether or not diseases can be eliminated without the use of prescriptions or long wait lists at the doctor's office. This system is an amazing gene editing tool that has been used for numerous experiments and has proved that it can be used to cut out certain strands of DNA in the genome that may cause diseases, infections, and mutations such as sickle cell, cystic fibrosis, and hemophilia. It's even been used for agricultural benefit. CRISPR-Cas9 was used to cut out diseases from tomatoes, wheat, and corn to produce healthy growing foods for people like us to enjoy. It's even been proved that yes, E. coli relies on HDR, or homology-directed repair, to repair its broken DNA. But it's been proved that it can also be reliant on NHEJ, or non-homologous end joining. This is proven by just using a different sequence of primers. It's also shown that it was tested in a lab in 2015 on human embryos that were created, and they cut out a heart disease from these embryos, and 72% of them came out mutation-free. It's also being studied to determine if it can be used for a new technique for cancer therapy. I believe that in due time, this system will help improve the way that modern technology affects how people apply modern medicine in everyday life. The purpose of this project was to tackle this concept to be able to give back to Red Rocks Community College students for the spring semester of 2021. I personally did not know very much about molecular biology before starting this project, so it gave me a chance to learn about this amazing technology and get to research everything about the positives and negatives of using this system. Originally, this project was more hands-on in the lab, using this technology to genetically modify fission yeast. But I'm glad that it turned into more of a remote learning process, because I was able to be given time to truly enjoy what I was researching. This project definitely was not easy. Learning molecular biology, learning how to buy equipment for the laboratory, and of course how to transpose these ideas and concepts into a course for students to study. The main purpose for creating this course is because if there's early exposure to CRISPR technology, this could help to demystify the growing field of genetics. This creates scientists and doctors that are in touch with modern techniques during their education. It's very important for students to hear about this technology early in their studies so that they are more in touch with how technology is rapidly advancing alongside modern medicine. Because maybe one day these students will be in the labs and in the hospitals using this technology to help people down the road. Thank you.